Hi, today we're going to look at ATP, which is the most important molecule on, to all cellular life on planet Earth. Here are your learning objectives. Take a moment and read through these. Now, living things can't eat ATP, but ATP is the preferred cellular energy currency, which you've probably heard a lot of in all of your previous biology courses. So, ATP is the preferred energy currency of cellular life. And it doesn't matter if you're a frog or a butterfly or a bacteria, the enzymes in these cells require ATP as the energetic molecule. And I'll say, right, it's the primary preferred energetic molecule within cells. Why ATP? Why all these cells? Because, right, life descends with modification from a single common ancestor. So the idea is that there's conservation in evolution. And as life evolved, right, so we have descent with modification and branching from this early common cell. And that the enzymes in that cell evolved to use ATP as cellular energy currency. And so because we have conserved genetics, and therefore conserved protein structure, all forms of life evolved enzymes that require ATP, right? And also manufacture ATP. Now, ATP has a dual life. One role of ATP is to serve as this high energy molecule that we use to push forward otherwise energetically unfavorable reactions in cells. And the other role of ATP is to serve as a nucleotide, which is incorporated into DNA and RNA. We have two sides of metabolism. We have catabolism and anabolism. Catabolism is primarily, it's written for you, an oxidative process whereby we extract energy or we liberate energy from the chemical bonds of foodstuffs like glucose, which we can eat, and we recapture that chemical energy in the bonds of ATP. The high energy bonds of ATP are broken to release energy that drives anabolic and general reductive processes. So we take small molecule precursors and we build them into large macromolecules. Now, as you can see in this diagram, we recycle the large portion of this molecule of adenosine triphosphate, and we're recycling the adenine purine ring, ribose, and the the alpha phosphate, and you'll see that coming up. We do this recycling of the bulk of the molecule of ATP, and we just continually phosphorylate, dephosphorylate, phosphorylate, dephosphorylate. Here we add phosphate, and that P with a circle is the shorthand I use for phosphate. And here, when we liberate the energy, we remove phosphate. So we've got this ATP cycle, and that ATP cycle couples chemical, right, or potential energy. And we have a release of energy that we can use to do some form of cellular work. And you're probably thinking about that definition of work where it's, the movement of a mass through a distance is work. Well, if we look at cellular work, we can define these types of cellular work. So transport of something across a membrane, and by something I mean a small molecule or protein that we transport across a membrane. We can even transport ions across the membrane with the aid of ATP. Biosynthesis, when you hear biosynthesis, think anabolism, anabolic pathways. We're building larger molecules from smaller precursors. Signaling, think binding of an extracellular item to the surface of the cell, and then we have essentially a phosphorylation cascade that relies on ATP. And we have mobility. So you could think about the movement of cells from one location to another. So just the easiest thing to think about is a flagellum. Okay, now, if you look at this ATP cycle, if we were to measure how much ATP your body uses in a day, about 65 kilograms of ATP is used in your body each day. 65 kilograms of ATP each day. So we really do need to recycle the bulk of the ATP molecule to meet the energy needs of the body. Synthesizing ATP. How do we make ATP? We are chemoheterotrophs, and so we synthesize ATP through carbon oxidation. So think glycolysis, TCA cycle, electron transport chain. And then we have a hydrogen ion gradient, which powers ATP synthase. So that's us, that's in people. I like to think about this as the drive train of metabolism. Okay, now photosynthesis, obviously that's not us, right? That's green plants, and then we've got algae, and we have photosynthetic bacteria. Sulfur oxidation, think bacterial world, and also think about our ancient bacteria, the archaea. It's all about the ATP. Every living thing that ever was or is has evolved to use primarily ATP to power enzymatic reactions within the cell. And again, there is a molecular basis for every biological action, every cellular action. ATP is serving here as the primary energetic molecule. If you can no longer make ATP, that's called the condition of death, because you need ATP to power your cells. And just to make sure that you're getting this convention here, since you weren't in Biochem 1, blue atoms are nitrogens, white atoms are hydrogens, the black atoms are carbons, the red are oxygens, and the orange here are phosphorus. So this is the standard convention, the standard color convention for the different atoms in molecules. And what you're looking at here is a ball and stick representation of ATP. This would be your adenine ring. Here's your ribose sugar, and this would be your triphosphate here. So the central core of metabolism in all forms of life on planet Earth is very highly conserved. And I like to think about this central core of metabolism like the drive train of metabolism, or right, the power train. So the main components of the drivetrain or the powertrain in a vehicle would be the engine and the transmission. And that general technology, having an engine that uses some type of chemical fuel, right? It's a chemical reaction, whether it's a gas-powered car or an electric car. There is a chemical reaction. So there's chemical energy that's liberated and converted to mechanical energy in that engine. And then we have to step down that mechanical energy in order to make the car go, right? So you need an engine and transmission. That technology is pretty standard in all cars that are manufactured. In living things, this standard drivetrain of metabolism is pretty constant. Why? Because of evolution. Nature is purposeful. Nature will continue to use and improve, most times improve, the same systems that have been time tested. So, most living things have some form of glycolysis within them. So, glycolysis is the pathway where glucose is broken into two pyruvate molecules, and that's going to be our first focus. So, glucose with 6 carbons is broken into 3 carbon molecules of pyruvate. So, see glycolysis, sugar breaking. That is glycolysis. And again, most living things can use some form of glucose and will have some form of a glycolytic path. Then you can see here that the Krebs cycle, aka the TCA cycle, or the citric acid cycle, right? This is found in many living things, but not all. And over here you can see we have the electron transport chain. This is the electron transport chain here. I know they look like little bacteria, but they're not. They're complexes within the electron transport chain. Most living things, I shouldn't even say most. Many living things have also the electron transport chain. And so there's a conserved approach here evolutionarily. Now, we can't use glucose to directly power cells because glucose is a very stable molecule. I mean, there's a lot of chemical energy in the bonds of glucose, but we need some way to access that energy and extract it into a common chemical form that we can use then to form high energy bonds in ATP. And ATP is like the standard molecule that is recognized by most enzymes that require an input of energy to move a chemical reaction forward. So you can see here some more common themes. We have oxidative pathways, right? These are oxidative pathways from here down. By the way, the TCA cycle completes the oxidation of pyruvate. These oxidative pathways give us high energy intermediates, which I call reducing equivalents. These reducing equivalents feed electrons and hydrogens into the electron transport chain. The electron transport chain then builds a hydrogen ion gradient, and the hydrogen ion gradient is used to run ATP synthase, which generates for us ATP. Do you see here chemiosmosis? Chemiosmosis refers to the movement of hydrogen ions through ATP synthase. Very similar to what we looked at just now, but now we're considering also the energy level of the different intermediates, the different biochemical intermediates in these pathways. So we have energy level on the y-axis, and on the x-axis we have progress of energy extraction over time. So this is essentially time. So glucose enters into glycolysis in the Krebs cycle, also known as TCA cycle. And here we're going to fully oxidize the carbons in glucose until we end up with fully oxidized carbon. This is fully oxidized carbon in the form of carbon dioxide. So there's 6 carbons in glucose. Once it's fully oxidized, we're going to have 6 molecules of CO2. So for every 1 glucose, we end up with 6 CO2s. In the process of glycolysis, we get 2 NADH out for each glucose that goes in. In the process of TCA cycle, we're going to get 3 NADH and 1 FADH2 for each pyruvate that enters into that cycle. So these reducing equivalents are going to go on to power the electron transport chain. And so you see here hydrogen ions with electrons, hydrogen ions with electrons. They are going to enter into what's along these arrows and is not shown, the electron transport chain, which builds a hydrogen ion gradient that is contained within the intermembrane space of the mitochondria. That hydrogen ion gradient is going to power ATP synthase. And so we get tons and tons of ATP that we can use to perform some cellular work. Here are some images from a microbiology textbook. So, you know, a lot of what we know about human metabolism, we know because these metabolic systems were studied in bacteria and yeast. So these microorganisms are very easy to work with in the laboratory. We can grow them quickly to high numbers, and we can harvest different proteins and enzymes from these organisms and use those proteins and enzymes in assays, right? Biochemical assays to understand better how they work together in metabolic pathways. There are different general approaches to making ATP. And here are the major general approaches to making ATP that we are going to consider. And at any rate, please focus on the terminal electron acceptor and the number of ATP produced. So in aerobic respiration, which is, I'll put most of our cells, but most especially brain and heart muscle, we will run glycolysis, TCA cycle, and then electron transport chain. This is the electron transport chain. And this thing here is ATP synthase. We use oxygen. Oh, I should erase this so you can see it. We use oxygen as the final electron acceptor. This is a very, very efficient way of making ATP. And so in general, 38 ATP are produced, and we call that aerobic respiration. Why? Because oxygen is used. So oxygen is aerobic. And then respiration, respiration means a lot of things to a lot of different specialties. If you're a clinician, respiration means breaths per minute. If you're a biochemist, respiration means running the electron transport chain. That is respiration to a biochemist. And I guess if you're a physiologist, we could talk about external respiration, which is gas exchange at the alveolar membrane, and internal respiration, which is gas exchange at the cellular membrane, right, within the cells that are consuming the oxygen. But to a biochemist, respiration means running the electron transport chain. So we're looking at all of these redox reactions, reduction oxidation reactions performed within the electron transport chain. That is respiration to a biochemist. So let's now look at anaerobic respiration. The approach here is very similar to the approach in aerobic respiration, except it is anaerobic respiration, right? Respiration, we're still running the electron transport chain, but in an anaerobic way. So there is no oxygen being used here. We're using it says non-oxygen electron acceptors, but I like to tell students that we are using an inorganic molecule that is not molecular oxygen, right? Don't say just oxygen because it's ambiguous. What do you mean oxygen? Oxygen the gas? Oxygen the element, right? No, molecular oxygen or diatomic oxygen. So either molecular oxygen or diatomic oxygen. Don't say oxygen. Try to be as precise as you can. So anaerobic respiration is an anaerobic process, meaning that oxygen is not used. Let's look at fermentation. In fermentation, we have no TCA cycle and no electron transport chain being used. In this case, we're running glycolysis fermentation. And it's rather inefficient in as far as we only get 2 ATP produced. Let me erase this, and I want to add in for you some more terminology. So how do we refer to these different cells that have different approaches to making ATP? If you are an obligate aerobe, then you must do aerobic respiration. If you are an obligate anaerobe, then you must do anaerobic respiration. If you are if you are an obligate fermenter, you must do fermentation. However, there's always got to be one fancy outlier, and we'll say that there are facultative anaerobes. If you are a facultative anaerobe, you have your choice. You will do aerobic respiration when oxygen is present, or you can switch to fermentation when there is no O2 present. So facultative anaerobes, E. coli is a very famous facultative anaerobe, so are yeast. In our bodies, skeletal muscle is a facultative anaerobe. If we think in our bodies, who's an obligate fermenter? Red blood cells are obligate fermenters.